Okay, in this video tutorial I'm going to look at what we call monosaccharides. So, before we get into that, just some of the basics. Living things are primarily composed out of these elements: oxygen, carbon, hydrogen, and nitrogen. We also contain small amounts of calcium, phosphorus, and sulfur. But really, the bulk of us is really just those four elements. In fact, if we look at their proportions, you can see here, most of us is actually oxygen. That's largely due to the water that's in us. Carbon, hydrogen, nitrogen, calcium, and then a few other little bits and pieces. So most of us is just a few of these elements. Now, when we talk about that we're made out of a few elements, most students have a good understanding from their junior years of the atomic level, so they know what atoms and elements are. They know what basic molecules are, so they know about H2O and carbon dioxide and things like that. They also, at this level, a lot of students sort of start to appreciate the organelle level, so that cells have these different parts to them, the organelles. But they kind of often lack an understanding of this intermediate, this group in the middle here, macromolecules. So all those organelles, those things, are actually made out of large groups of what we call macromolecules. A macromolecule being just a really big molecule, a large group of atoms assembled usually in a slightly more complex way. So this is really, in terms of our levels of organization, how we get from atoms being pieced together all the way through to an organism. It's the macromolecule level that people often have the least understanding with. So we're going to look at some of the macromolecules. Now the nice thing is that essentially there are patterns to how these things form. We can classify them into groups, and that makes it a little bit simpler. It's not just millions of totally different types of macromolecules. So just like when you learnt about the atom, you looked at the periodic table and how they're arranged. When you started to look at basic compounds, you looked at bonding and, probably electron configurations and valence shells and things like that, to look at the rules about how they connect together. There are some general rules or basic principles about these macromolecules which can help us understand sort of the different types or how they're organized. So we're going to look at that, and so I'm going to start with monosaccharides as a good example of one of these macromolecules. Alright, so building blocks of life. These are the ones that the next few video tutorials are going to cover. So we're going to start with monosaccharides, which can be pieced together to form polysaccharides, which are just long chains of many of these monosaccharides, mono meaning one, poly meaning many. Organic bases can be used to make nucleotides, and those are used to make nucleic acid, which maybe you might already pick up on that DNA is deoxyribose nucleic acid. So these are the building blocks of DNA. We're not going to look at the bases overly. It's worth noting that monosaccharides are one of the building blocks needed to make DNA or RNA, so those forms of genetic information. The organic basis side of it, we might look at that more when we sort of cover genetic control. Amino acids make up proteins, and fatty acids and glycerol, which make up lipids. So these are essentially the building blocks of life. So you can already see, you probably would have been thinking there are millions of different types of macromolecules, but we can actually sort of classify or group them into the major ones and look at the most important ones, and often they're quite similar. So of all the different possible combinations, there seems to be some patterns or trends in terms of there are common groups or types that are quite similar, and that's essentially what we're looking at here. So let's start with our monosaccharides. Alright, so name this molecule, C6H12O6. Well, some of you would have covered it as juniors when you did photosynthesis, so you might know this as glucose. So this is a carbohydrate. Now, the reason I name this molecule is actually, if you follow the same naming rules, some of the rules you learnt as juniors, you should be able to name it even without any knowledge of from the actual formula or having memorized it yourself. So normally if a carbon has a metal, now this one doesn't, but often we start with, but in this case we're going to start with carbon anyway. if it contains, we put that name first, and then the other element, and if it contains oxygen, the ending changes to 8. So, you've probably looked at things like copper sulfate, CuSO4, copper, sulfur, and oxygen, so it becomes copper sulfate. So this ending here, if we look at it, carbon hydrate, carbohydrate. So that's actually just how this molecule is named, carbohydrate. Carbohydrate. So anything that's comprised of carbon, hydrogen, and oxygen is most likely a carbohydrate. So this includes things like sugars, starch, glycogen, and cellulose. And we're going to have a look at all of those, but in this tutorial we're going to focus primarily on the sugars. So, another name for sugars, monosaccharides. So our glucose, C6H12O6, goes by many names. We can call it a carbohydrate. So that's probably one of the more general terms we can use. We can call it a monosaccharide, meaning mono, single, saccharide, stems from I think it's the Latin for sweet. So single sweet or single sugar. So it's a single ring sugar. So glucose is a monosaccharide. So we've already got a few different names for it. We can call it a carbohydrate, a monosaccharide, or most specifically glucose. So monosaccharides or carbohydrates generally have this formula, CH2O. So if you think about that, it's actually carbon and water. And that's really, if you, when we break down a lot of carbohydrates, we break them down using oxygen to release carbon dioxide, combining it with this carbon here, and the other byproduct is water. So our carbohydrates are really just a complex combination of carbon and water, where the bonds between the carbon and the hydrogens actually hold a lot of energy. So there are 3 types of monosaccharides that we're going to consider. Triose ones, which are 3 carbons long, pentose, 5 carbons long, and hexose, 6 carbons. So glucose, C6H12O6, is a hexose, a 6 carbon sugar. So again, we've got another name for it. We can call glucose a carbohydrate, a monosaccharide, a single ring sugar, a hexose, a 6 carbon sugar, and most specifically we can give it the name glucose as the particular arrangement specific to, that's the name of that molecule. So monosaccharides, single ring sugars. Alright, why do we need monosaccharides? What are they important? What function do they serve? So firstly, they're an energy source. So in our bodies we convert a lot of foods to glucose, which circulates through our blood, and that basically reaches our cells, and then our cells will convert the glucose to ATP. ATP is basically cellular energy. That's what cells run on. Now ATP is when we use ATP, it gets converted into ADP. So ATP stands for adenosine triphosphate, and it gets broken down into adenosine diphosphate, and actually a free phosphate is released. So triphosphate versus diphosphate, and it's that's how we utilize energy from ATP, by breaking it down into ADP. All you really need to know here is that basically glucose is a source of energy that often or monosaccharides are often a source of energy that our bodies run on, and cells often basically run on glucose or these monosaccharides. And use them to generate ATP, which is cellular energy. However, monosaccharides are also used as building blocks for materials like starch, glycogen, and cellulose. Starch and glycogen are basically just energy stores. So plants store energy as starch. We store a bit of energy as glycogen, but we also store it as fat. And cellulose is a large component of the cell wall in plants, so structural in that case. It's also an essential component of RNA. So RNA contains a ribose, a pentose sugar, and DNA deoxyribose nucleic acid also contains a sugar. So if you've covered the structure of DNA or RNA, you know there is a sugar, a single ring sugar, a monosaccharide in the backbone of the DNA molecule. And it's also even a structural component or part of ATP, that's cellular energy. So monosaccharides, basically, you probably don't need to know all that detail, but it's important that you appreciate that monosaccharides are an important source of energy for us, and they also act as a building block or material from which we can make stores of energy, but even biological molecules that are crucial to our function. So things like DNA and ATP, that's cellular energy. So, glucose, a hexose sugar. So hexose just means 6. Hex, like hexagon, alright, hexose is a 6 carbon sugar. So its molecular formula, C6H12O6. If we write out its structural formula, it actually looks like this. Now note the numbering. We can number the carbons starting at oxygen. So basically I like to think of this oxygen with the double bond here as 0. And then carbon number 1, 2, 3, 4, 5, and 6 down the bottom. So if we look at these, basically each carbon, the first one's got just the O, the double bond O, but all the others basically have an OH group on either side and a hydrogen on the other side. So all the others have an OH and a hydrogen. OH, hydrogen, OH, hydrogen. Alright, right down to the end here, where this one actually has 2 hydrogens and an OH, just to, I guess, cap off the end. So largely, again, think of this as we've got carbon with water, an oxygen and 2 hydrogens kind of attached to either side of the carbon. And that's partly where carbohydrates get their name. Carbohydrate, hydrated carbon. So note that numbering. That's going to be important when we look at the structure in a little bit more detail. So these straight chain sugars aren't often really found in large quantities in real life in nature. And that's because they can fold up to form these ring structures. So here we've got the straight chain, and what happens is this double bond here opens up and it bonds to carbon number 5 down here. So carbon 1 in this case, so this is a straight chain version of glucose, carbon 1, this double bond opens up and will bond onto essentially carbon 5 via the oxygen. What happens is that the hydrogen is basically transferred. We get an OH group coming off carbon 1 in place of the other bond. So you can think of it as this OH group jumping onto carbon 1 and the double bond opening up and connecting onto carbon 5. So again, the OH group here jumping onto carbon 1 and this double bond opening up and connecting via this oxygen to carbon 5. So if we look at the ring here, that's exactly what we get. Carbon 1, alright, now has an OH group that it didn't have before, alright, and is connected via that oxygen, no longer a double bond, but connected via that oxygen to carbon number 5. So this is glucose as a ring structure, a hexose. You notice it's actually a hexagon, alright, although one of them is actually oxygen in here. So these are the ring structures of glucose, which are more commonly found because they're slightly more stable. So there are 2 different versions of the rings that can be formed, and that just depends on how this molecule, what position it is when it binds, when you get that bond forming. So here, the OH and the H's can essentially rotate around. They can swap sides. You've got rotation around these bonds. But in the ring structure, that rotation is restricted. So what we have is alpha glucose here, you'll notice. Now, firstly, you'll notice there's 2 different ways we can draw this. Here we've drawn in all the carbons, all the hydrogens, we've basically drawn in every single atom and shown each bond. Now, just to simplify it so it's a little bit clearer and a little bit tidier, what we can do is remove the carbons, so they're just shown as a corner or as a join. So there's a carbon here and one here, there's another carbon here, and we don't show any hydrogens. We just assume that any spare spaces So carbon tends to form 4 bonds. If there are any free spaces, so like this carbon here has got 1 to the oxygen, 1 to the next carbon, and 1 to the OH, that there'll be a hydrogen filling that last space. So here we simply show the carbons as the intersections, so we don't even draw on the carbons, and then we show basically just the oxygens and the OH groups. So that's probably it's just a little bit tidier and a little bit simple, a little bit quicker to draw. And you can see here there are 2 slightly different versions of glucose. And they're very similar, and it's quite easy, guys, to really remember the difference. There's a little bit of a trick. Looking at my numbering, starting at oxygen, 0. Nice, nice coincidence there. Oxygen, 0, circle. Alright. Carbon 1, carbon 2, 3, 4, 5, and 6 sticking up the top. So carbon 1, 2, 3, 4, 5, 6 sticking up the top. So that's how they're numbered. If we look at alpha glucose, the first one on carbon 1, we've got the OH coming down. On the next one, we've got the OH coming down as well, and then up, down, and on the last one, up, on that carbon number 6. Number 5 doesn't actually have an OH group, okay? And that's because it's that point in the ring where that extra carbon joins or sticks up. But anyway, it means that in terms of our OH groups, we actually have a pattern. So starting at carbon 1, we go down, down, up, down, up. So we start with in the down position, and the first 2 are both in the down position. For beta glucose, the only difference is the OH on carbon 1 is the other way. It's facing upward. So for beta glucose, which is an optical isomer of glucose, we've got the OH in the up position, then down, up, down, up. So on this one, it actually just alternates as you go around the ring. Up, down, up, down, up. So alpha glucose, down, down, up, down, up. So starting down, down, and then alternating. Beta glucose, up, down, up, just alternating, starting with the up position. Okay, so guys, you are expected to be familiar with these structures because some of the features here are quite important in terms of how they can combine or link together, and that's what we're going to look at in the next tutorial. So it does pay to make yourself familiar with these structures. Okay, alright, so we're going to make a little bit of a start on this one. We're going to start talking about how they can combine. So monosaccharides can be joined in a condensation reaction. Now a condensation reaction is just a reaction that produces water, H2O. So if we look here, we've got a monosaccharide, so that's a single ring sugar, and another monosaccharide, a single ring sugar. In this case, they're both alpha glucose, so you can see here down, down, up, down, up. Alright, and on the other one, down, down, up, down, up. Okay, so 1 and 2, those OH groups both in the down position. What you can see happens here is that the OH from on carbon 4, for instance, actually you can get this reaction occurring between any of the OHs on any of the carbons, but some are more common than others. But when the OH group from one combines with the hydrogen from another, essentially water is generated, and then that oxygen acts as a bridge. There's now a bond connecting the 2 sugars together via that oxygen. So you can see here pulling water out, pulling this hydrogen, this oxygen, and this hydrogen, the OH and the H group out to make water, and this bond on carbon 4 will now connect onto the oxygen here, alright, which is also connected to carbon 1 on the other. So that can actually form between any of the OH groups on any position. In theory, but some are much more common than others. So in this reaction going this way, we pull water out, so water is produced. If you want to drive it the other way, adding water is going to be essential to drive this reaction back the other way. Alright, but that's usually an enzyme-driven process as well. So, the OH or the hydroxyl group on one sugar combines with the H from another. That's the key thing, and that's called a condensation reaction. And that's how we can link sugars together. Now when we link sugars together, we get what's called a disaccharide. So di just means 2, mono meaning 1. So we had monosaccharides, single ring sugars. We've now got a disaccharide, a double ring sugar. So guys, disaccharides are still considered sugars. However, if you piece together more than 2, once you get 3, 4, 5, or long chains of them, they're no longer considered sugars. So monosaccharides and disaccharides are considered sugars, whereas polysaccharides are not considered sugars. So when we use that term sugar, that's what we really mean, a mono or a disaccharide. Okay. So that's it for monosaccharides, guys. So this little picture here, my little presentation, if you look at that, that's supposed to be glucose there. That 6 carbon hexagon shaped sugar little molecule with the alternating the hydrogens and the OH groups around the ring. So this one here, if you look carefully, is supposed to be alpha glucose. We've got an OH group down here, OH down, so down, down, and then up, down, up the top. Okay, so I haven't actually put in any of the letters, but just a pictorial kind of reference to the glucose molecule there. So guys, I hope that was helpful, and in the next video tutorial we're going to look at polysaccharides, so how we can piece these together into longer chain molecules.